So anytime you see P-H-O-R-E in a science word, that means to hold up or to bear. Okay, to hold up or to bear. And so it's just holding up the Antheridia a little bit up into the air. And same with the Archegoniaphore. So again, it's just up on that stalk. And so it's that stalk is called the Archegoniaphore. And then right at the tip of that, you'll see where the egg is getting produced. All right. And so they're trying to show a blow up of the tip of the Antheridia where you have the sperm. And they're showing free water again, need free water to get the sperm to splash over to the egg. And this is interesting because the Archegonia are kind of hidden underneath. It kind of looks like a drooping umbrella with the Archegoniaphore, and the eggs are way up underneath. It's kind of surprising that you can get the splash of the sperm just right and get that in there. But again, you can still see, a single egg in the Archegonium. One Archegonium has one egg. And they're showing lots of sperm got splashed over. So hopefully one of those made it there. Sperm and egg unite and you get your fertilization.
So, the ANTHER, does that mean to hold up?
No, P-H-O-R-E. The P-H-O-R-E, for means to hold up or to bear, if you look up that part of the word. So it's just saying this, so this little stalk, I'll show you some better pictures. This little stalk with the, well, I can't reach it. This little stalk with the Antheridia on the top, that whole thing, you'd say that's an Antheridiophore. And then you'd say, and here's the Antheridia in the, so they're zooming into the Antheridia tissue there. So they think Marchantia and similar, because they're so low to the ground, maybe that's just a strategy to try to get better fertilization to occur where the splashing can allow for a little bit better aim into where the eggs are located in the Archegonia. And then the egg, once it's fertilized, then we say it's a zygote and it just goes through lots of growth through mitosis until we get the sporophyte. I'll show you some pictures of those. And then here they're trying to show there's little streaky bits of tissue here, just kind of long, thin bits of tissue. And those are called elaters. And they're just helping, they think, with spore dispersal, because when they dry out, they kind of give this twisting action as they do that. And they think it helps kind of get the spores out after they've gone through meiosis. Okay, so same basic idea with you have your gametophyte as the dominant phase, the sporophyte is less persistent over time. And that same idea of fertilization and meiosis to get you back and forth between the sporophyte and gametophyte phase. So even though we focus on Marchantia and the flat thallus type, the leafy liverworts are actually more common. If you get a good sample of the thallus type liverwort and Marchantia in particular, it's pretty good at forming gemma cups pretty consistently. So that's what you're looking at here, those cute little cups that are sitting on top of the gametophyte thallus body. And then if you look in, this is a little bit better picture, and you can see little tiny bits of tissue in there. And so you can imagine, you're looking down on it, so you can imagine water dropping right into that with rain, and you'll get the splashing of that cloned tissue out. So this is a gemma cup under the microscope with the gemmae are in the purple there. So that's just cloned tissue. So once that splashes out of there and lands somewhere suitable, it's going to develop into the gametophyte. So that's all asexual. And so they're good at doing that. Here are some pictures of the Archegoniaphore on the right here. So they kind of look like a shredded umbrella, like it's been through a windstorm. The Archegoniaphore. And then here, an Antheridiophore is a little bit more flat and looks more like a typical umbrella that you might use Here's one where you're looking down on the gametophyte that has the antheridiaphore. So you can't see the stalk here. But here's a really nice image showing the antheridiaphore. So you'd say taken together, oh yeah, look at all those antheridiaphores. And then you have to zoom in to the top part that kind of looks like the top of the umbrella to find the antheridia, which is being shown here under the microscope. So all that dark blue tissue is going to be tons of sperm in each antheridium. And then you got to wait for water to come along and splash and hopefully get the sperm to the egg. So now we're looking at the archegoniophore with that more shredded umbrella look. And actually what you see this thing peeking out here, that's the sporophyte. So the egg underneath there got fertilized. These are some antheridiaphores down here, but let's zoom in on these. You can see this kind of tannish brown tissue start. This is the best one right here, more mature. So you have this egg underneath there in the archegonium got fertilized, and you see the sporophyte starting to peek out of there. But it doesn't get very big. The sporophyte, it doesn't get very big like what we see with mosses, and it's pretty sheltered there. It's hard to see. You would need a nice sharp eye or a hand lens. This one has several sporophytes that are peeking out there. All right. So you can, again, if you can find the sporophyte, you know that's where the egg was, because once the egg gets fertilized, you get the zygote, and then the zygote develops into the sporophyte. So you know that's the female gametophyte there. And so you see lots and lots so it's amazing that, there was enough rain splashing going on where you got all those eggs fertilized, and then you get the development of the sporophytes. So here's a nice blow up of that. Under the microscope, it would look like this. So this is the egg already got fertilized. So the egg was right around here. And then it got fertilized by the sperm, became a zygote, and then went through lots and lots of mitosis and differentiation to give you that sporophyte. So that's the little petite sporophyte. Hard to spot. You definitely need to look closely. All right. We looked at this idea last time too with the aquatic liverworts. That actually looks more like a moss, maybe. A lot of people are fooled by that. They just say it's a moss, but it's not. It's a liverwort. So that's probably what most people have experience with in terms of liverworts. They're not super common, at least in this area. They're not super commonly found, and not huge populations, but you can buy them in pet stores if you want to get oxygen in your aquaria in a more natural form. And that's just a really pretty picture, again, by an expert photographer with small, petite, microscopic things where you can get some really nice detail. So very pretty. Okay, and then the last group is the hornworts. Again, these, not very many of them still existing today. They're hard to find. In fact, I think when I go out hiking, it's hard for me to find them. It's not easy. There's only, as far as I know, Anthoceros, that genus is the only genus that's temperate and native to this, more temperate regions of the world, like around here. There's only about 100 species total. And they kind of look like a thallus liverwort, but they have a more rounded appearance when you look at the gametophyte. And everything's kind of embedded into the tissue. They don't have any fancy stalks that hold up, or, the Antheridia and Archegoniophore is just kind of all right there. And they are co-evolved often with cyanobacteria, nostoc. And nostoc is very famous for its ability to fix nitrogen. So that is, not only can it do photosynthesis, but it can fix nitrogen. So that is a nice partnership to have. Definitely benefit from the nostoc's ability to do nitrogen fixation. And then nostoc's also doing photosynthesis, and the hornwort can provide a nice kind of stable home for the nostoc. So the hornwort is called a hornwort because it's got this big, long horn that sticks up off. So that's the actual sporophyte. And it is very persistent when you compare like all the sporophyte phases. The hornwort really can have a nice long phase of a sporophyte phase because it's got actually a meristematic region that allows it to keep growing over months. And so it doesn't just grow once, stop, and then slowly die. It tends to refresh itself with new growth. And so it actually greens up. Because it persists for so long, it can do photosynthesis. It has chlorophyll. It does photosynthesis. I'll show you a picture of this. It also has elaters, those long, skinny, flat pieces of tissue that help with, they think, with spore dispersal. And so it's just this long, slender horn. And then when the spores are ready to go, it just kind of splits down the middle and kind of fans open, and the spores get released with the help of the elaters. So, it's kind of impressive once it gets into the sporophyte phase that the sporophyte can last so long. It's interesting to see that. So here you can see the gametophyte phase is more of a flat, more roundish structure. And then so somewhere in this area, the eggs were there and got fertilized by the sperm. And then that zygote developed into the sporophyte phase. And here they're showing this one is starting to split down the middle longitudinally, and it's releasing the spores. So they're showing it green because it does persist over time. It can hang out for months and do photosynthesis. And so I'm not going to put any visual like this on the exam because this is hard to see, but you're actually looking at the Antheridia here. These are very close up, nice imagery, microscope imagery. That's still Antheridia there. So all those little bumps are going to develop into sperm. And then here you have Archegonia. Again, these are not going to show up on any kind of visual because it's hard to tell what's going on here. But let's say one of these eggs got fertilized. So then you'd start to see this coming out. So, you had a fertilized egg that developed into the zygote, and now you're starting to see the sporophyte develop from that successful fertilization. And then this image is trying to show, it's definitely green. They're showing the meristematic region. So this area just keeps producing, consistently will divide and produce new cells if needed. So they're long-lived phase. Here, this is what it looks like in a more natural setting. So if you see that around here, you consider yourself lucky. They're hard to find. You have to go a little bit more damp, consistently damp areas. But I have a hard time finding them. This is under the microscope. This is just a section of the hornwort. And all the purpley pink, those are the spores that are going to get released. And then here they're trying to show that in the underside, that's where you tend to see the nostoc hanging out and fixing nitrogen. Again, I wouldn't ask this visual. Here under the microscope, if you take the gametophyte and look at it under the microscope, if you're lucky, you can find the Anabina. So here too, you have right here, it's again hard to see cells here. This is kind of like the cavity area here. And well, this is pretty good. These cells here, that's all cyanobacteria that is hanging out in the cavity, pretty protected little home doing nitrogen fixation. Okay. This is kind of a nice synopsis to get you comfortable with the phyla names and who belongs to which phyla and some of the key characteristics. If I haven't specifically mentioned something on here, don't worry about it. So just think about what I talked about in the previous slides. Some of this, there's a few things in here I wouldn't ask you about because we didn't really talk about. Okay, so what I thought we could do for the rest of the time is go over the exam material and kind of get you geared for that. And the next topic that will start next week is shifting from the bryophytes into the seedless vascular plants. But that obviously is not going to be on this exam. So let's think about the exam. Oh, let me go back. So remember, leaves part 2 is where we're going to start. Okay, that's all looking at how they modify that tissue to do certain things, like, tendrils, or got the Venus flytrap and more exotic adaptations. So don't forget about that slide set. And then after that, we got into tissue culture and biotechnology. I didn't talk specifically about plant breeding. That was a homework assignment, but it's just a very short set of slides. The chapters or the section I want you to read is pretty short. And then fungi, we talked about endophytes and mycorrhizal fungi, lichens. Then I wanted you to refresh, if you haven't had soils, a little bit about soil. If you've already had the soils class, that's a pretty easy section on soil. You got way more in your soils class. And then algae and the bryophytes. Okay, so thinking about like with the leaves. I think this stuff is pretty straightforward. Most of these images are just backing up what is being shown in slide 2 of all the different ways leaves can adapt. So then I give the visual examples of all of that. So either being able to recognize something very obvious or talking about something in the essay portion would be something I could see would show up on the exam. And then we didn't get very detailed about secondary metabolites, but understanding what secondary metabolites are in general, and that it's part of their defense mechanism. And then thinking about economic importance of leaves, all the many different ways that humans use that. I think it's very important to think about and be ready to talk about or recognize, so that you get a better appreciation for how much we use plants in our everyday life. Any questions on this slide set? Oh, and I added the Canadian flag. I can't believe I forgot about that. Duh. So, yeah. Any questions on this slide set? Yes.
I have a question. I think on slide 27. What are the flowers? Yeah, the one where the flickers on. What are the
Oh,
flowers?
yeah, those are lily of the valley. Very cute. Yeah. They smell lovely. It reminds me so much of my mom. She loved lily of the valley, and she would plant those around in our landscaping. But once we got dogs, we couldn't do that anymore because if they chewed on it. So But yeah, very lovely plant to have, but you have to be aware it's poisonous. They're very pretty to look at. They're very low to the ground, give off a marvelous scent. But yeah, they're poisonous. Any other questions? So leaves, and then we got into tissue culture. So that one is a little bit more technical. Tissue culture. So tissue culture, that was thinking about the idea that if you know what you're doing, you can pretty much take any living part of the plant and clone it. As long as you got a good recipe technique that you can follow. And there's more and more of those now that are available. And that's because plants, they're just amazing at how they've decentralized their functioning across their whole body. So you can chop them up and take tiny bits and pieces of them and still get them to grow into a full complete plant. So that totipotency is pretty amazing. We can't do that. The human body is very centralized. We have very specific tissue that does very specific things. And if that gets damaged, we're not going to survive very well. So we talked about tissue cultures and some of the methods employed in that, some of the difficulties, because you always have endophytes to think about and worry about. So either you get your tissue culture medium with some kind of antimicrobial in it to try to slow their growth. Surface sterilization is an issue. But if you can successfully do that, and you don't get overrun by endophytes in the process, you can actually clone a plant. And then after we finished the tissue culture idea, the basics of that, we got into some of the biotechnology used in order to transform plants, which just means you're putting foreign genes into the plant in some manner. Usually you have to do that with tissue culture techniques so that you can get the entire plant to be cloned. That's usually the goal. You want the entire plant transformed. And so you tend to work in tissue culture, and often in those cases, you tend to work in liquid broth, where you can agitate and get the plant cells kind of floating around so that when you introduce your gene, especially with agrobacterium, you can just mix the agrobacterium in there and they'll do their thing. So agrobacterium is such a wonderful workhorse, so to speak, because you can use it. You can put your gene of interest into its plasmid and then just let it be what it naturally does, and it will insert its TI plasmid into the plant chromosome. So agrobacterium is used heavily because it's pretty broad host range. And then you get the one thing you have to do is somehow determine which cells got transformed. And so you usually have to have some kind of marker, either an antibiotic marker, or you can expose the cells to whatever you're trying. Like if you're like, I want this plant to be herbicide resistant, well, then you expose it to the herbicide, and whatever makes it, that one got transformed. So you have to have some way to detect that you did this successfully. You got a transformed cell, and then you start cloning it in tissue culture. So this has come such a long way in the last 10, 20 years. It's really amazing what they're able to do now with the biotechnology and introducing. So agrobacterium is really common for that, but if you can't use agrobacterium for some reason, then you have to resort to other methods. So we looked at some of those, including a gene gun, but there's lots of options. So if you like to play with gadgets, you could work in a lab like this. But it just depends on your plant and which protocols have been worked out, which method might be the easiest. So be familiar with these methods. And You know, we did talk about some of the more bizarre options like planted bodies and protoplast. No, definitely know what protoplast means. That's going to show up a fair bit in this section. You got to know what that is, otherwise you're going to get lost. So protoplast is just the idea of you're stripping the cell walls off. You're keeping the cell alive. It's got an intact cell membrane, but there is a protocol that usually with special enzymes that you use to carefully strip off the cell wall. So we talked about protoplast fusion and somatic hybrids, artificial seeds. So know those ideas. Those are going to be on the exam too. Questions on any of these concepts? Yes.
Yeah, I just have a quick question on like tissue culture. So if you have like endophytes occurring, is that going to just like ruin the tissue culture fully or can it?
Not always. They can,
Okay.
like with personal experience, I've seen endophytes pop up, but they stay in a small amount of growth. Like this one, I've had situations like this when my plant is growing and there's like a smattering of endophytes. And I'm able to like take this out very carefully and get rid of all the agar and all of that and then transfer it and it makes it. So it just depends. It's not necessarily, oh my gosh, it's all ruined. I gotta start over. Obviously,
Yeah.
these are toast. But I've had many situations where there's just a smattering of endophytes and I'm able to get it. And, you know, I've even kept like where it showed up. But yeah, okay, the plant definitely has endophytes coming with it. And I just keep it to see what's gonna happen. And the endophytes never get out of control. So there's some kind of dynamic going on there where maybe the plant is keeping the endophyte more in check. Kind of fun to watch over time. Other questions on anything in the slide set here? Yeah. So yeah, there's some terminology here that need to think about. So if you can't breed the plants to get the trait you want, so that's what that first bullet point means. It's actually incompatible germplasm. That just means you can't get them to have sex naturally. They're different species, so by definition, they can't have sex with each other and produce viable offspring. So then you could do the protoplast fusion idea where you can get protoplasts from different plants and then very carefully combine them in a tissue culture, usually a liquid media, and you can get them to fuse. And then that's the idea of creating a hybrid. So you say it's a hybrid fusion of these protoplasts. It's a hybrid fusion of these protoplasts. And then basically it is once they grow and develop, you would say, you know, oh, well, yeah, that's a somatic hybrid. It came from two different plant species. And so once they've grown up, then you tend to use the word somatic hybrid. That's how that plant was created. It was through hybrid fusion, and this plant is a somatic hybrid. So that's usually how you talk about this. So when you fuse the protoplast, you say, oh, from different species, it's a hybrid fusions from those protoplasts. And then if you can take those and grow them up into a fully grown plant, then when they sell that plant, they would label it as a somatic hybrid. So you probably are going to see that terminology showing up more and more for when you go to buy certain plants, if they disclose that idea. And then when you hear the word or phrase somatic embryo, somatic is body cells. And so they're saying we created this embryo using body cells, not sperm and egg. And that's bizarre. But again, for some plants, they've been able to do this where they can take just bits of the plant cells from the starting plant, put it into tissue culture, and they got a recipe down where they can encourage those few cells to form a cluster that's like a developing embryo. And I've never done this. And I'm pretty sure I wouldn't be able to get my hands on the actual recipe. That's probably proprietary information that they don't want to say anything. But amazingly, they can get these body cells, not sperm and egg, and they can coax them into looking like and behaving like an embryo that will then develop into the plant. So you will see for sale artificial seeds. If you see that, then that's what they did. They will create these somatic embryos and then encase them in this gelatinous matrix to make it physically protected and then sell it. So you may see the newer term is seeds. So anyway, you'll see that for sale. Synthetic seeds are artificial. I think they're stepping away from artificial seeds because maybe some people kind of have a negative thought about that. And seeds is a little bit more hip, I guess, so we'll go with that.
Yeah.
So you'll see more about that. Definitely. I've seen some of those for sale when I'm shopping for seeds and certain companies, so. Pretty amazing. Other questions in this slide set? So there's a fair bit of terminology in this slide set and thinking about, processes, the tissue culture process and inserting genes into plants process. So then after that, we got into the fungi, I believe, right? So with fungi, I really only focused on the symbiotic idea. So we looked at fungal endophytes, just a little intro into that. So just like you have trillions of cells, microbes that hang out with you, plants now we're realizing have that as well. Any living organism is well colonized by microbes because we stepped into their world. When we evolved, it was their world first. And so we're well colonized by microbes on and in us, so are plants. And so there's a lot of interest in what are they? Who's there? How did they get there? What are they doing? Can we use them for anything that we want to solve any problems or help with anything? So it's a very active area of research, drug discovery, and just so many new papers getting published on all the things that these microbes are doing for the plant and helping the plant adapt. So we got into endophytes, looked at some beautiful fungal plates of endophytes, and then we got into mycorrhizal relationships. We really only narrowed in on the ectomycorrhizal fungi and then the endo or the VAM type fungi. I'll definitely be asking not only about endophytes, but mycorrhizal fungi. So know the difference between ecto and the endo type. For sure, I'm going to be asking several questions about the fungi for sure. So do you have any questions on like how they associate with the plant? I think it's a good idea to do compare and contrast when you're studying.
Mycorrhizal fungi, they are associated with like root culture, right? Like they're roots of like.
Typically the roots. Yeah.
And like share between different roots that are.
They can, an ecosystem. Yeah, they're like a underground. They call it what the world wood wide web. I think something like that instead of world wide web, wood wide web. So yeah, very much, very exciting research going on with how they are driving ecosystem dynamics.
So enterprise, they also like do like get into kind of roots, but like they go into some membranes.
Well,
Okay.
so none of the fungi actually penetrated cell membrane. They hung up to us. Cozy with it.
can't penetrate through.
So what,
Under the soil.
what that is like specifically like the difference between like.
Well, an endophyte, the way I define an endophyte is it lives its life internal into the tissue of the plant. I don't consider mycorrhizal fungi endophytes because they have so much of their hyphae out from the plant. So they live partly in the root tissue and a whole bunch out, and so I don't define that as an endophyte. And so, but if you look at ectomycorrhizal fungi versus endo,
You see some, like with slide 18 here.
this is an ectomycorrhizal fungi. And it's growing in between, the hyphae go in between the cell walls.
Of the roots. And that's where they're limited to. But with endomycorrhizal fungi, so slide 26, they're a little bit more intimate in the sense that they can make it past the cell wall of the root cells and plug up to the cell membrane, but they never penetrate. So even though it looks like they're in the cell, you'd have to get an electron microscope to show there's actually a plant membrane around all of that. And so the fungal hyphae never gets physical contact with the cytoplasm. So that's a key compare and contrast between the two major types we covered. Yes,
Oh, in between, the cells, correct, for the
ecto. Mhm.
yeah, ectophytes. And then the endos, they're able to go in between the cell wall and the membrane.
Yes.
Okay.
Yes, yep. So if you think about, like, if this hand's the cell membrane and this is this, the cell wall comes out to here, they can get in between and they can kind of smoosh up to the cell membrane. And it, sometimes they'll kind of push in or against it to create that it looks like it's inside, but it's really not. It's never making physical, the hyphae never swims around in the cytoplasm.
Like when you kind of like push your hair, it's like a food lining cover, just fills it in, but you're not pushing.
Yeah, that's a good analogy.
Okay, I don't remember my.
Yes.
Sorry.
No, it's okay. So,
Okay, so for the endophytes, do they provide like structure by chance, like being like smashed in between, do they provide like extra structure at like?
Are you talking about endophytes or endomycorrhizal?
Endophytes, because they're in between the cells, right?
No, endophytes tend to just be in the tissue, like entirely. Yeah, so they're never,
Okay.
yeah. So,
Yeah, and then we're actually.
yes, so endomycorrhizal fungi can go into the root tissue, they can make contact with cell membrane. The endomycorrhizal fungi also have hyphae that go out into the soil. That's one of the key benefits of the plant, is that they have so much fungal hyphae going out into the soil, we're really good at that. Helping the plant get access to water, minerals. Yeah, but endophytes live their entire life inside the plant. And they can go anywhere. They don't have to stay in the root tissue. Even if that's their entry point, they can go up the stem and into the leaves and into the flowers. I mean, they can go everywhere, into the seeds. So they're not limited to the roots. How many people have taken soils class? Okay, so the section on the soil, I'm pretty sure you had most of that in soils, I would guess, the information on the soil system idea. But I don't know how much they get into how microbes help release minerals, but definitely that's really important. When you are a botany person, you need to appreciate that you need a really healthy soil system. And when I say healthy, I mean a very nice levels of microbes in the soil, good amounts of fungi and bacteria that can help the plant.
You want us to understand like the Liotropic series idea?
The basic idea, yeah, just the basic idea that they have worked out which minerals can easily bump off other minerals, so to speak, so that cation exchange idea. You have, that's definitely going on all the time in soil systems. You get cation exchange, as long as there's water moving through the system, you're gonna have cation exchange happening. And when you get your soil tested, they'll give you a number for your cation exchange, and the higher the number, the better, pretty much. So you always should look at that, because that will tell you a lot of how your plants will fare in that soil system. Are they gonna have an easy time, or is it gonna be challenging? Other questions? Yeah. Annabelle? Okay. Lichens, we got into those. So I did say no, these, for genus names, please. So make sure you do. And then just the basic idea of who's there. And, a little bit of the biology. Questions on lichens? So once spring rolls around more fully in this area where we have more snow melt, definitely peel your eyes for lichens. They're everywhere. Just got to train your eye for it. Well, then we got into algae. I think we did this one. All right. I can't remember if this is working. Yes, Annabelle.
So.
Uh-huh.
Mentioned, we talked about cyanobacteria, we like touched on it, like,
No, I'm only going to focus on the algae in here for the exam. I like throwing the cyanobacteria into lab because I'm partial to microbes, so I throw it in there for this section, but for the lecture exam, we'll just focus on the algae. So again, the phylum names going to be important. Knowing what each phylum name is corresponding to, anatomy, or in morphology, so that slide 2, definitely you're going to be asked about those ideas. And then this slide 8 gives you the phyla I want you to know. And obviously we're not getting into any of their sexual reproduction, but definitely their ecology, and uses. Think about their uses. I think that's really important. Algae get short shrift in terms of being paid attention to for that kind of thing. And I think they're going to become more of a thing, especially for you as your generation moves forward. You're going to probably be eating a lot of algae in the future.
This might be a really goofy question, but are algae plants?
Oh, no, that's not a good question. I told some people to follow that. So, no, I say no. Some people say yes, but they're in their own.
Green algae is like, oh.
So I have adjacent, but I don't.
Because I was thinking of them like in photosynthesis and like all the plant stuff, they don't have like vascular like tissue or like plant.
Right. I mean, plants evolved from green algae or the charophytes, but don't really consider them plants. They're their own wacky little group. All right, I'll be checking email. If you need to email me a question, you can do that.